will make you host so that you can.
Okay, I'm hoping that everybody can see the screen now.
Yes.
Terrific. So what I was going to focus on in this talk today is the differences between mouse and human when it comes to the brain, especially the cerebral cortex. And we've been looking at early stages of development, progenitors that produce neurons, and they are, of course, part of the glial lineage. They turn out to be radioglial cells. And this is just a schematic of over 20 years of work on rodents that give us some idea of the lineages of the cell types in the adult brain, starting out with neuroepithelial cells that convert to radioglia. When they start making neurons, they produce neurons that migrate radially and eventually populate the cortical plate. And then at the end of neurogenesis, the radioglia transform into astrocytes, different type of glial cell. And we're applying similar principles now to understand human brain development. And just for comparison, I think one of the obvious differences is the progenitor population is more diverse, and development occurs over a longer period of time, and there are many other complexities. And I was going to focus on some of these over the next 45 minutes or so. So I realize not everyone here is a developmental neurobiologist, so I just want to highlight that if you look at a cortical cross-section at early stages of rodent, like mouse development shown here, there's a ventricular zone lining the ventricular cavity, shown here in green are the stains for radioglial cells. And they produce daughters that are the intermediate progenitors that form another layer, this so-called subventricular zone above. And these two progenitor regions, the ventricular zone with radioglia, the subventricular zone with intermediate progenitors, together actually make all the rest of the cells that you find in the adult mouse brain. At early human stages, it looks very much the same. There are radioglial cells and intermediate progenitor cells that are lineage related to each other. But what happens over the next few weeks in human development is this huge expansion of the subventricular zone into another region that's been referred to as the outer subventricular zone, shown here on the right. And these different markers and colors that you see indicate stages of cell cycle. So these are cycling progenitors in this huge progenitor zone that doesn't exist in the mouse. And you might notice the ventricular surface down at the bottom, which is the main region of neurogenesis for rodents, is becoming very, very small at a stage when the outer subventricular progenitor region is very, very big. And these are very different features between human and mouse, of course. So a number of years ago, we started looking at, and others started looking at what kind of progenitors might produce neurons in the human outer subventricular zone. And I want to highlight one particular cell that we found that shows a morphology on the left that looks like a radioglial cell. It has a long radial fiber, which supports neuronal migration. But the cell body is not at the ventricle. It's not an epithelial type cell. The cell body is actually away from the ventricle in this outer subventricular zone with no contact to the adhesion belt where the other neuroepithelial and radioglial cells sit. So it's very distinct in that regard, but it's stained with all the markers we had at the time for radioglial cells at the ventricle. But morphologically, as I mentioned, they were very different. And so we gave them a different name. We called them outer subventricular radioglia-like cells, or ORG cells for short. And they've subsequently been referred to as basal radioglia by Wieland-Eutner, but I'll refer to them as outer radioglia for the rest of my talk. So we're lucky at UCSF that we get donated tissue that we can actually section and slice culture. And in this case, there's a cross-section of human developing cortex showing the outer subventricular zone, where we've selectively labeled only these progenitors that I referred to, the outer radioglia. And they're highly radial, they migrate, and they proliferate as they move up toward the cortical plate. Occasionally, you'll see cells moving the opposite direction. We now know that those are oligodendrocyte precursors, which I won't really refer to much in this current talk. But the behavior of these cells when they divide was really very unique. And it's highlighted in these images where we only look at a handful of the cells. And if you watch one of these cells when it divides, it jumps and divides. The cell body translocates along this radial fiber in M phase, just before cytokinesis occurs. This is a behavior that hadn't been described in any other cell type before. And so we called it Mitotic Somal Translocation, or MST for short. And early on, it became a defining characteristic of these cells. So within another few years, we put together a sort of work in progress schematic of what we think goes on in human cortical development. The radioglial cells, like in the mouse, shown here at the bottom, produce daughter intermediate progenitors. And they divide once, most of the time, producing a pair of identical neurons that migrate into a position in the cortex. This happens over and over again, about 11 cell cycles in the mouse. But in humans, some of these cells, these radioglial cells, delaminate from the ventricle in a kind of mesenchymal epithelial transition, or epithelial mesenchymal transition, I should say. And these outer radioglial cells then jump away into this outer subventricular zone, and they go through a series of asymmetrical divisions, each time producing a daughter cell here, which is not so much an intermediate progenitor as it is a transit amplifying cell, because it divides multiple times to produce a clone of neurons that then migrate to the cortex. And over time, they layer inside out and form this radial column of cells that derive from one or more of these neighboring outer radioglial cells. At the end of neurogenesis, we think both the ventricular radioglia and the outer radioglia transform into astrocytes. And we have evidence now that there may be different classes of astrocytes in the human brain. So one of the tools that we and others have been using to try to disentangle the cell composition is single cell RNA sequencing. This I'm sure most of you are probably familiar with now. But we first adopted this relatively early on using a microfluidic chip shown here diagrammatically at the bottom, which could capture only up to about 60 cells at a time. And so our first published study involved what now is a laughably small population of about 300 cells. And now we're doing millions of cells at a time, as are others. But at that time, the technology really wasn't that far advanced. Drop sequencing hadn't emerged. But even with a small number of 300 cells sequenced at a single cell level, we could cluster them according to their cell types shown here. And we could come up with genes that are uniquely expressed by different cell types. And that gave us tools which we didn't have before. And I just want to highlight on the left genes that we found associated with ventricular radioglia that hadn't been known before. And then on the right, more interesting to me, our genes that we discovered are highly enriched in the outer radioglial cells. And these are confirmed in situ hybridization studies shown below, where you can see these marker genes are expressed very highly in the outer radioglial cells. And we validated this both morphologically by showing double labeling of these ORG-like cells, and through behavior by time-lapse imaging these cells that are jumping and dividing, and showing that they also express these genes, these proteins. Sorry about that. So this allowed us to look at the cell types during development in human cortical slices. And one of the conclusions we drew was this sort of revision of what we think happens during cortical development in humans and primates. So the early stages, when the deep layers are formed, those neurons are produced by ventricular radioglia through a process that looks very much like what happens in the mouse. But around the middle of corticogenesis, when the middle cortical layers are being formed, there's a huge increase in the number of outer radioglia that develop from the ventricular zone by jumping away. And from then on, the upper cortical layers are almost entirely produced by the outer radioglial cells, not the ventricular radioglia. And interestingly, the ventricular radioglia transform into these short process cells where the fibers don't regrow to reach the peel surface way up here, but instead they make contact on blood vessels. And these we now call truncated radioglia. And they have a different gene signature, a different morphology, and we think they have a different lineage than the other ventricular and outer ventricular radioglia. So we think they're a distinct cell type, which means that we've now defined three classes of radioglia in the human developing brain: the ventricular radioglia, the outer radioglia, and these truncated radioglia, which I'll talk about a little further as we go along. So having a gene profile that identifies these outer radioglia allowed us to look for them in other species and also in this case in human organoids. Some of you are probably familiar with organoids. I'll be talking about them because they're a model of human brain development. So we and others have been growing organoids in the laboratory, and we started doing single cell sequencing and noticed that this fingerprint or gene expression pattern for outer radioglial cells emerges in the organoids after about 10 to 15 weeks in culture, suggesting that these organoids actually contain outer radioglial cells. So we sectioned the organoids and slice cultured them just like we did the fetal tissue. And if you look at this cell, which has the morphology of an outer radioglial cell, shown here in a looped image, it jumps and divides in a characteristic behavior with the same features, distance jumped and so on, as in the fetal tissue. So we actually do have outer radioglial-like cells growing in our organoids. That allowed us to look at whether they were disease-specific features related to the cell type. And at the same time, I had a postdoc Marina Berstein in the laboratory, who was interested in this disease, lissencephaly. Now, in rodents like mice and rats, the brain is smooth, as shown down here in a human. But in humans, this is only present, of course, with a very severe malformation. Normally, the human and primate brain, most primates, is highly folded, as shown in an MRI scan up above. But in this disease, the brain actually is smaller. There's a larger ventricle and it's microcephalic. But most distinguishing feature is the fact that the folds are missing and the cortex is very smooth, which is why it's called lissencephaly. And this is a major cortical malformation. We were able to obtain skin cells from 3 patients with this very severe form of what was known as Miller-Dieker lissencephaly. And we compared their organoids grown from these 3 lines to normal lines. And early on, the development in the organoids looked pretty similar. There were some differences, but the biggest difference we saw was in outer radioglial cells. And we saw that in the wild-type organoids, they jumped and divided just as they do in fetal cells. But in the Miller-Dieker lissencephaly organoids, these cells jump much further, 50% or 100% larger, twice as big a jump. But then having jumped, they arrested in mitosis. Some of them didn't divide at all, and others divided only over many hours instead of minutes. So this is a defect in the outer radioglial cells in this disease, which actually I think very nicely links this cell type to brain enlargement or expansion, and also cortical folding, which we think both depend on the cells that are generated during these outer radioglial cell divisions. And this was quantified, obviously. It also showed us a phenotype that had been missed in mouse models of Miller-Dieker lissencephaly, because of course the mouse is, among other things, lissencephalic, as I mentioned, and doesn't contain these kind of outer radioglial cells. So the genes that distinguish or define outer radioglia are not just marker genes. They're clustered into very specific pathways that are features of the cell type. And they fall into 3 main categories shown here. They're involved in extracellular matrix production, in the epithelial to mesenchymal transition, which is how the cells originate, and renewal or stem cell maintenance pathways. And all of those networks have been described in the literature in this disease, a cancer that you're probably familiar with, glioblastoma multiforme, a very severe, untreatable tumor that doesn't occur in developing brain. It develops usually in patients who are in, they're decades old, they're 40 or 50 years old. So we were able to get primary samples from our neurosurgical colleagues and do single cell gene analysis. And here again, we found the signature for outer radioglial cells in the most aggressive or invasive forms of glioblastoma, suggesting to us that there might be ORG-like cells in these tumors. So we got fresh samples from the OR, the operating room, and we sliced and cultured them as we did our fetal tissue. And if you look at this cell in a tumor, which has the features, the morphological shape of an outer radioglial cell, if you watch the way it divides, it jumps and divides exactly like our fetal cells do. And so this suggested to us that there really were ORG-like cells in these tumors. And we used single cell analysis to compare all of the progenitor cells that could be cells of origin for this tumor. So here are the tumor single cell sequencing data on the top, and down below are our fetal data categorized for the different progenitors that have been speculated to lead to these tumors. And that includes adult astrocytic fibrous astrocytes, protoplasmic astrocytes, oligodendrocyte precursors, developing precursors of the OPCs, radioglial cells in early developing brain, and those intermediate progenitors I mentioned. But the highest correlation between the tumors and the progenitors in the fetal brain were in the radioglial population. And if we looked aside from just marker genes at networks, modules of gene expression that characterize these tumors and compare them to fetal cells, we found many of the same genes, these hub genes that define this cell type. One of these markers, HOPX, is widely used now as a marker for outer radioglia, and also PTPRZ1. This is a highly enriched cell marker for these radioglial cells in humans, but it's even more highly expressed, shown here in these violin plots, in the glioblastoma cells. So this is the expression level for this gene in glioblastoma, and this is a level in developing fetal radioglia, outer radioglia. But in the adult human brain, it's almost non-existent. So it's a really particular gene, and we think a gene network, that's enriched in these outer radioglial cells. And we were really struck that at the same time we were looking at this, there was a paper published just a couple of years earlier, which took advantage of the fact that PTPRZ1 is highly enriched in the most invasive tumors and knocked it down in xenografts in mice. And in the control mice, when they injected these tumor cells from human samples, they grew over 3 weeks quite significantly in the brains of these mice. But if they knocked down PTPRZ1 before they injected the tumors, there was either very slow growth or, as shown down below, in some cases, no growth at all, suggesting that PTPRZ1 is involved in the invasive or growth mechanisms of the tumor. And this is leading to a clinical trial now for pharmacological inhibitors of PTPRZ1. And in our tumor samples, we were able to enrich 2 fractions, one enriched in PTPRZ1, which we think contains these ORG-like cells, and a fraction that was depleted of these PTPRZ1 cells. And we injected those into organoids, normal cerebral organoids, which turned out to be a very good culture media for these glioblastoma cells. And so as shown here, a few weeks later, we had lots and lots of cells from the original PTPRZ1 population that were growing and distributing around these organoids. We then fact sorted, enriched for them, and showed that they actually produced cell types that matched the original diversity in the tumor sample. And so this gave us comfort and suggested the ORG-like cells are a glioblastoma stem cell population, capable when they're enriched in reproducing the same cell diversity that you find in the tumor itself. We don't think it's the only tumor stem cell, because we also found many different cell types in the adult tumors that grew out of the depleted population. But we do think it's an important cell type, and especially in the most aggressive forms of glioblastoma. Now, since our original studies, which were based, as I mentioned, only 300 cells, we've expanded these significantly in stages. So our next study, shown here, was with over 4,000 cells, also sequenced the 60 or 70 at a time using the Fluidigm platform. But it gave us a much larger population of cells to look at and to subcluster. But in addition to just using single cell sequencing for clustering, we looked at gene networks that I mentioned earlier, weighted gene co-expression analysis that gave us coherent modules of gene expression that really gave us more insight into the networks and gene networks that were activated in individual cell types that differed from each other. So we looked more carefully at my favorite cell type, the outer radioglial cell, and found quite a number of interesting gene networks that were very specific to this cell type during development. And one of them that we were especially interested in is the LIFR STAT3 pathway. And shown here, the SOX2 cells, which are in red, were highly enriched in this pathway, shown here in blue. But the daughters of these cells, the intermediate progenitors, shown here by EOMs or TBR2 staining in yellow, lacked any of this LIFR signaling pathway at all. So it was interesting that this self-renewal pathway, which is found in other stem cell populations and other organs, in the human developing brain is only found in the outer radioglial cells. And it's a pathway that leads to self-renewal when it's activated. So we wondered if this could solve a problem we had with our organoids, which is shown here. HOPX in red are the outer radioglial cells in the organoids. And they're there, as I showed you earlier, but in much lower numbers than you'd find in developing human brain. So we wondered if we activated the self-renewal pathway that's selective to this cell type, if we could expand their numbers. So we added LIF, which is the activator of this pathway, in our organoids. And in several weeks, we saw a huge increase in the number of outer radioglial cells in the organoids. And then a few months later, we saw a larger increase in the upper cortical layer cells that are the daughters or the cells produced by these outer radioglia. And so this is a way of sort of tweaking or improving, we think, our organoid populations. And it's a nice example, I think, of how you can use clues in normally developing brain to, in this case, modulate and improve the quality of this in vitro model of brain development. And one of the other pathways that was highly enriched in outer radioglea is the mTOR signaling pathway. And these are different effector genes in that pathway. And I won't go into the details of the way we mapped it here, but this is another way of expressing the fact that these genes are enriched in outer radioglial cells. And we confirmed that by looking in slices of human brain, staining for a gene known as PS6, the protein of which is a good marker of activated mTOR signaling. And it's shown here in red. It highlights selectively the outer radioglial cells. And so this also showed us that mTOR signaling was uniquely enriched or expressed in these cells. And that's of interest to us because the signaling pathway, the mTOR signaling pathway, has been implicated in diseases like autism and tuberous sclerosis and macrocephaly. And the enrichment in this cell type in humans suggests that this cell may be involved in the mechanisms of those diseases. And I'll touch on that at the end of my talk in just a little while. So we looked more carefully at what mTOR was doing in these radioglial, autoradioglial cells. And we used an inhibitor, rapamycin, of mTOR signaling. And compared to the controls on the left, we saw that the radial fibers, and these are mostly the autoradioglial fibers at this stage in human development, were missing in our gross cortical sections. And a more close analysis showed that not only were they missing, but they were truncated. They were much shorter. They didn't grow as long. That's quantified here and could be rescued if we activated mTOR signaling. We looked more carefully at dissociated autoradioglia treated with rapamycin. And the rapamycin not only shortened the primary process, but it caused this abnormal growth of multiple additional processes out of the basal region of these autoradioglia, where there's usually only 0 or 1 fiber at the most. So there was a major disorganization of the cytoskeletal structure of these autoradioglial cells. And to make a long story short, this is part of the pathway that activates actin fibers, and we think that this is disrupted when you block this mTOR signaling pathway in autoradioglia and leads to a distortion of the cytoarchitecture and neuronal migration as well. So I'll touch on that disease again, as I mentioned, later on, so hopefully you'll all remember those features. But now I want to depart for a minute and talk about these organoids, because they're being highly used all around the world now as models of human brain development and models for studying diseases. And we were in I think, a maybe unique, but certainly a very strong position to compare these organoids to primary tissue. We've been looking at the composition of normally developing brain, and now we have an opportunity to compare that to the cellular composition on a molecular level in these developing organoids and see how well they really reproduce normal brain development. So for those of you who aren't familiar with brain organoids, they're a little different than, say, intestinal organoids. There are organoids like the intestine, where you can take an adult intestinal fragment, you can extract the progenitor cells, and they can reproduce the gut, essentially, because they continuously renew that part of the organ throughout life. But when it comes to organs like the brain, you can't do that. You have to begin with a pluripotent-like cell and recapitulate developmental stages in order to form these cerebral organoids. And that takes time. It essentially takes the human gestational period of 9 months just to mature an organoid to the level of a sort of a newborn brain or newborn cerebral structure. So let me just highlight how they compare in these images. Here I've shown primary sections of developing human brain compared to sections of organoids stained with the same markers at roughly comparable stages of development. And the organization is very different, as you can see. The organoids have these rosettes that form where the progenitor zones are not linear as they are in normally developing brain. Instead, they're circular, but they have rudimentary organization. They have apical basal polarity within these rosettes. They undergo interkinetic nuclear migration when they divide, and they produce their daughters, which migrate away from these progenitor regions. And then over time, these rosettes disappear. Things look a little bit more chaotic, but you can sort of cherry pick areas that have some laminar organization, shown here in week 15, where the outer radioglia and radioglial cells, the progenitor cells, are shown here in red, and the neurons in blue. And that kind of resembles what happens in normally developing brain, which is, of course, much more exquisitely laminar, more beautifully organized. But this is a kind of rudimentary organization that resembles what happens in normal developing brain. And then our single cell sequencing was a good way of sort of deconvolving the composition of these models to see how well they compare to each other. So this is what we compared our organoids to. This is a Disney plot showing about how many cells? 189,000 cells, which we collected rather from periods across the neurogenic second trimester of human brain development. So we put all these cells together during cortical development in this large map, essentially. We then highlighted here in each of these panels genes that are associated with the major cell types, which include the radioglial progenitors, the outer radioglial progenitors, the intermediate progenitors, and then neurons, young neurons, excitatory cells, more mature excitatory cells, and the inhibitory cells, which are also found in the developing cortex. So with this as a sort of a blueprint, we compared them to similar studies done with a similar number of cells from organoids grown at periods that resembled those human developmental stages. And looking at the same genes I just showed you, in blue, you can see that we found the presence for all these major classes of cells, but the relative proportions were very different. So the young progenitors in ventricular radioglia and the young neurons shown below were pretty well represented. But the outer radioglial cells, the intermediate progenitors, the more mature neurons, as well as inhibitory cells, were far less well represented. So the proportions of these cells were very different. And I highlight that because many people are studying diseased organoids and looking at one of the phenotypes they look at is the proportion of cells in the organoids. The second thing they often look at is the sequence of maturation. And both of those features, the proportions of cell types and the sequence of maturation, are not the same in organoids as they are in normally developing brain. Now, what if we look at individual cell types and compare apples to apples? In our fetal samples, we find that our cell types that can be identified by individual genes indicate that there are around 600 genes that are expressed selectively in individual classes of cells. We do the same kind of analysis in organoids, and we find there are only 46 cell type defining genes. So the diversity, the rich diversity of gene expression in organoids is much less than the highly rich diversity that we find in normally developing brain. And if we compare which genes define which cells, the number that are actually shared is much smaller. And that's also expressed here, where we look at the diversity of cells in different groups. For example, excitatory neurons shown on the left, much higher diversity of excitatory neurons defined by gene expression in normally developing brain than in the organoids. In fact, in the organoids, the diversity is reduced to pretty much just two classes, the upper and deeper cortical layer cells. And much of the other rich diversity of cell types is missing. So these become kind of generic excitatory neurons in organoids rather than the real diverse types that you see in normally developing brain. And there may be another even bigger problem shown here. We look at gene expression that classifies two classes of cells, the radioglial cells and the excitatory neurons. And in developing brain, these are not shared. So shown down here in yellow on the bottom are the radioglial genes, which are not expressed by neurons. And conversely, here are the neural genes, which are not shared by radioglia. But in blue, we look at the same genes in the organoids, and we see that they're blended. In other words, radioglial cells in the organoids are expressing neural genes, and neurons in the radioglial cells and the organoids are expressing radioglial genes, things that we don't see in normally developing brain. Now, if we look at modules of expression instead of individual genes, we see there's a much better correlation across cell types between organoids and primary tissue, which is what's shown here in this upper series of graphs. So the radioglial modules, the intermediate progenitors, excitatory neurons, and inhibitory neurons, if you look at the modules of gene expression that are enriched in these cell types, they resemble their primary counterparts, which is good news. And if we look at the hub genes, which are shown down below, we find many of the same hub genes that define these cell types in developing brain are also enriched in the same cell types in organoids. So that's very good news. The glass is therefore half empty or half full, depending on how you look at it. But if we look at the modules of gene expression that really define organoids and not primary tissue, they're not random. They actually fall into very interesting pathways. And the one that struck us the most was this pathway. It's a cell stress pathway. And in the violin plots shown here to the right, you can see high enrichment in many of the genes associated with cellular stress in the organoids, across all our organoid cell lines. Compared to primary tissue, which are the first two violin plots, this is an extremely enriched cell stress pathway. And not only do we see cell stress, endoplasmic reticulum stress, but the glycolysis network pathways are also highly enriched. So the ER stress and glycolysis stress pathways are highly upregulated in these organoids. So the organoids are under a huge amount of metabolic stress compared to their primary cell counterparts. And that's not just the organoids that we grow in our lab. If we look at all the published single-cell data sets of organoids using different protocols, which are listed here on the right, they all show the same finding to different degrees: increased cellular stress in the organoids. So we think there's something about the way they're cultured that induces stress. And if that could be improved, we think the fidelity of the cell types would also be improved. And what's shown here below is that this doesn't improve over time. So that young organoids or old organoids, they all show essentially the same elevated stress pathways across time. So this suggests that you should be careful when using organoids to model diseases that might be affected by cellular stress. And that includes most neurodegenerative diseases, where cellular stress is part of the feature of that disease. So since we started our study of single-cell RNA sequencing, we've expanded in two developmental time directions. We've looked at earlier stages and we've looked at later stages, which I want to highlight now. So the earlier stages are the stage of transition between neuroepithelial cells to radioglia, which is when neurons begin to form. And there are several features that define this transition. One of them that we noticed a little while ago is the mechanism of cell division and the morphological and dynamic features, which are shown in these time-lapse images of human neuroepithelial cells undergoing division. And if you look at an individual cell, like this one, for example, or the one next to it, the radioglial cell fiber retracts, and then the cell divides, and then both daughter cells regrow their fibers. And that's a symmetrical morphological division. And we think it reflects symmetrical divisions, which are shown here in individual images. So a single radioglial cell, or in this case, neuroepithelial cell, retracts the fiber, divides, both daughter cells regrow their fibers, and they do this over and over again as the population increases. This is what's known as the founder population. But they change their behavior, as I showed you earlier, when neurons are produced. At that point, the single fiber never retracts. The cell divides and the daughter cell migrates away, but the parent cell does not retract the fiber. And that's a characteristic of radioglial cell divisions. And it's very different than neuroepithelial cell division. So we knew there were at least those features that distinguish the cell types. But now we had molecular signatures to associate with this transition. And what's shown here in a sort of dynamic mapping are individual genes that are highly enriched in neuroepithelial cells and go way down when they turn into radioglia. And conversely, there are genes that are very lowly expressed in neuroepithelial cells, but then become highly expressed as they transition into radioglial cells. And just to show you one of the validations, the gene single-cell gene networks that I'm showing you on the left for Notch1 is shown in blue, highly expressed in neuroepithelial cells, almost not at all in radioglia. And if you look at the blue staining panel here for Notch1, in early neuroepithelial cells, it's not present. In neuroepithelial cells, as they increase in number, you can see it highly enriched. But then as radioglial cells appear and the neuroepithelial cells transition, it disappears again. So this is validation of the pathway dynamics that we see in our single-cell data sets. So we now have genes that are enriched in different neuroepithelial cells versus radioglia. How well are they conserved across species? So just looking at one of these genes, C1orf61, shown here in red, we find that it's expressed in our primary human data, as I showed you, in neuroepithelial cells, but then it disappears when they turn into radioglia. If we look at our organoids, we see a similar pattern. We see expression of this gene when they're at early stages of neuroepithelial transition into radioglia, but once they become radioglia, it disappears. So this is one of the genes that's nicely conserved in our organoids. We also had chimp organoids and see the same thing. So it looks like it's well preserved in primates, not just in humans, and in organoids, but not in mouse, as shown here. This is a gene that is not expressed in neuroepithelial cells in mouse. And it's just one example of genes that we find that are unique to human or to primate in our data set that we find not expressed or not convergent with mouse. And we've also looked at cell types according to what we think are not cell states, but truly distinct cell types during this transition. And we define 9 progenitor subpopulations across the developing cortex that I'll get into in a moment that we think are distinct. And these are in the radioglial cell clusters. And we're always interested in diseases, as I've highlighted a little bit during my talk. And if we look at some of the genes that are associated with risk factors for diseases like autism or schizophrenia, we're struck by how many of these genes are enriched at this very early stage between neuroepithelial cells or early radioglia, suggesting that even though many of these diseases don't manifest until after birth, they may originate in changes in the cell populations very, very early on. And the other thing we've been able to do with these very early cell populations is begin to look at how the aerial map arises. And for those of you who work on other organ systems, I should mention that the brain is really specific because the function of a neuron, the same neuron, will depend on its location. So a layer 5 pyramidal cell in the prefrontal cortex will have a totally different function than that same cell type in the occipital cortex, at the back of the brain. And so we're very interested in how that occurs. How does the aerial difference in terms of location emerge from what begins as a apparently uniform neuroepithelial cell layer? And so we can begin to look at that by our individual RNA sequencing data from cell types dissected in different regions across these early developmental stages. And so shown here are these graphics known as constellation plots. And they show by these lines the association or similarity between cell types. And shown here are the radioglial cells in brown. And at these different brain regions, like the prefrontal area, the parietal area, motor area, visual area, which are shown here in different circles, the association of the genes among each other is much more similar than across their daughter cells, which are the intermediate progenitors, shown here in brown, or the neurons, shown in blue. What this tells us is that the radioglia have a lot more in common with each other across these regions than they do with the neurons or the intermediate progenitors in those same brain regions. In contrast, the intermediate progenitors that the radioglia produce, which are shown here in brown, are much more closely related, and that's what these lines indicate, with the brain regions, for example, the visual cortex at the back of the brain or the prefrontal areas in the front of the brain. And they're more closely related to the neurons in those brain regions, like the visual area or prefrontal area, than the radioglia are. So this is a little bit complicated concept, but this is another way of showing that. So here we show different genes are shown schematically on the left. They're not individually indicated. Oh, sorry. I'm close to being finished, but not quite. So let me just mention that what we see here across time from left to right is that the genes in the front of the brain, that's what the prefrontal cortex is here, or at the occipital back of the brain, which are the V1 visual areas here, those genes remain pretty similar across development. And that supports the idea that genes that define these two areas, the front of the brain, the back of the brain, are a map that remains similar across time. And as you move from the ventricular zone where the progenitors are to the neurons, which are shown here on the right, those same gene signatures define the front of the brain and the back of the brain. But in between, which is what's shown here, you can see all of these swerving lines. The genes that define the temporal region in the beginning of neurogenesis actually shift so that halfway through neurogenesis, it's a whole different set of genes that are now defining this piece of brain. And then by the end of neurogenesis, it's shifted yet again. So they're very dynamic changes in the genes that define these regions over time. That's not the case at the front or the back of the brain, where those genes remain constant. So this supports what's known as the Proto-Map Hypothesis, suggesting that the neuroepithelial cells have a map of the cortex early on. And that as development progresses, what happens is that map stays the same. It just gets translated into the developing radially expanding cortex. So that concept turns out to be true at the front and back of the brain. But in the middle of the brain, it's not that. It's something else, which is known as the proto-cortex hypothesis. And using spatial transcriptomics, which is shown here on the bottom panels, we've looked at the spatial distribution of the genes that define these regions, which confirms the big differences between the front of the brain and the back of the brain, and the fact that these aerial genes start to emerge in between at later stages. And that's a schematic that we show here. Early on, it's the front and back of the brain that are most different. Whoops. But then as time goes on, these differences remain, but the other areas in between, the motor areas, somatosensory areas, parietal areas, become progressively more and more parcellated. And we think that occurs as neurons mature and may be plastic and dependent on interactions between these cells and maybe the thalamus, which is connecting to the cortex at that stage. So we're getting some insight into the mechanisms of arealization by looking at our single cell data. And this now has to be confirmed maybe by using organoid models in vitro. And the final thing I wanted to end with is evolution, which I promised in my title. So we and others are very interested in what makes us human, what is different in brain development in humans compared to non-human primates. And to make that comparison, finding out what's human specific requires an examination of our closest living ancestors, which are the chimpanzees. But unfortunately, we don't have access to fetal chimpanzee developing brain. We do have access to macaque, which is an out group, as it were, to look at evolutionary changes. And things that we find that are shared by chimpanzee and other monkeys like macaque, but not in human, will be human specific. So we have to look at 3 species at least of primates in order to make a claim for a human specific feature. So how do we do that? Well, I had a developmental biologist who was also an evolutionary biologist. Alex Pollan joined my lab a number of years ago as a postdoc, and he tackled this problem. And he did this by growing organoids from multiple human individuals, 10 of them, and multiple chimpanzees, 8 different chimpanzees. And you need to do that in order to make sure you're not looking at just genetic background differences. So he had different sexes, different individuals, and he developed it took a while, a protocol that worked equally well to grow organoids, which are shown here, from all of these species, human, chimpanzee, gorilla, and macaque. And so this shows organoids in humans on the top and chimpanzee is on the bottom. They're very similar in their structure. They matured at slightly different times, but we were able to look at them in terms of their composition, single cell composition. And the good news is that we were able to show that you could, in our bioinformatic plots, mix the cell types from fetal human, fetal macaque, organoids from human, and organoids from chimpanzees, and define all of the same cell types. So we could then, with this as a background, compare apples to apples. We could look at the same cell type across species at the same or comparable stages of development. So that's a really rich data set, which I'd invite any of you who are interested to take a look at, and you can ask questions of that data set on your own. But we were, of course, very interested in rated glial cells. So with this data set, we were now able to look at which genes define radial glia across these different species. And shown here in these 2 violin plots are genes that are highly enriched in fetal human radial glia, in radial glia from human organoids, but not in fetal macaque, shown here in pale blue, or in chimpanzee organoids, brain organoids, shown here in blue. So these are genes that are human specific, and not only that, but they're cell type specific. They're enriched in human radial glia, but not in other non-human primate radial glia. And that's also true for this gene, TMEM3033. We also found genes that show the opposite pattern, that are enriched in other non-human primates, radial glia, but not in human radial glia. So these are genes that were lost in human evolution in radial glial cells compared to non-human primates. Not only can we look at those individual genes, we could look at networks of gene expression. And so I want to end with this, which is the mTOR pathway that I showed you earlier is enriched in outer radial glia in human. So here again, in fetal human, I'm happy to say that this genes shown here on the right, which are part of the mTOR signaling pathway, are enriched in primary tissue as our earlier data suggested. They're also enriched in outer radial glia from human organoids, which matches very nicely the fetal data, which is very reassuring. But what's really interesting is that we don't see mTOR pathway enrichment in the fetal macaque or in the chimpanzee organoids. So we can now add to our understanding of this pathway, not only that it's enriched in human outer radial glia, but it's a human-specific enrichment that isn't matched in non-human primates. So if this pathway is important as we think it is for diseases like autism, it suggests that you not only can't study that in a mouse, but you can't even study it in non-human primates. You have to use a human model, which shows this enrichment in order to really understand how this pathway is involved in diseases like macrocephaly and autism. So I wanted to end with these conclusions. that there's a greater diversity of neural stem cell subtypes in the human than you find in the mouse and even other related non-human primates. That cell type specific genetic programs we find in the human progenitors in particular are not present in the mouse. And that the organoids, which can model some of these human specific features, can be very powerful, but you have to be careful because they're not exactly the same. And one of the differences I mentioned is that they have stress pathways that are very activated and that they have degraded cell type identity. And that could be improved if we could understand how to grow our organoids better. And then one of the things we're starting to look at are developmental features in humans, like, for example, this Proto-Map that I mentioned, which gives way to proto-cortex as cortex develops, and diseases may actually express themselves during these transitional early stages. And that's something that we and others are now starting to examine. So that was a very quick overview. I wanted to highlight the people in my lab who have contributed to this project, and in particular, these two very gifted postdocs and one MD PhD student who's worked very heavily on the single cell RNA sequencing, and especially the work we've done comparing human to organoid. And our funding sources, and especially the NIH Brain Initiative, which when it was started was the Obama Brain Initiative. That's been a terrific help and a continuing source of funding for our projects. So that's it. I want to thank you for your attention, and be happy to answer any questions that may have come up.
Some questions? Alejandro.
Hi, thank you for the talk. A lot of data, a lot of projects. So I have two questions from mainly the organoids. I'm interested in asking you. One of them, how is the interneural migration in these organoids, and whether do you think this could be a source of difference with the human development? And maybe in the same question is, do you think if by adding microglia, at least for later stages, would somehow compensate part of the development? I don't know, things you could add to the system. Right.
So just let me see if I understood. When you said migration, did you say interneuron migration or?
Yeah, I think that so you're looking at the radial migration, right, from what would be the cortex, and in development, interneurons come from other places. So is something like this happening also in these organoids?
Yeah, that's a very interesting question. Let me start with that. So we've been growing what are called directed organoids. We're using a protocol tweaked from something that Yoshiki Sasai first introduced, which really creates dorsal cortical organoids. So they resemble the cortex, or they're sometimes referred to as cerebral organoids. The problem with interneurons is that they don't originate from the dorsal cortex. They originate from the ventral pallium, or ventral forebrain, an area known as the ganglionic eminences, mostly from the medial ganglionic eminence, which is not present in the dorsal cortex, a different structure. But those interneurons then migrate into the cortex during normal brain development. So some people like Sergio Pasca at Stanford have been making what they call assembloids, which are directed dorsal organoids and directed ventral organoids. And they fuse them together and allow the interneurons to migrate from the ventral to the dorsal. And that's a way of reproducing to some extent what happens during normal development. Most people are not doing that. They're using the protocol similar to ours to make dorsal organoids. And if you watch those long enough, you'll see that they do make interneurons, even though they're not supposed to, or they wouldn't in normal dorsal cortex. And the other interesting difference is that you have excitatory cells produced first, the deeper cortical layer, and then the upper cortical layer cells. Then you have interneurons. So you have to wait pretty long to start getting a reasonable number of interneurons in your dorsal cortical organoids. That's very different than what happens in normal developing brain, because birth dating studies using thymidine, years ago done by Pascal Rakich and others, show that the same layer interneurons and same layer excitatory cells are born at the same time. They're born in different regions, but they have the same birth date. So in normally developing brain, you don't see a delayed maturation of interneurons. The interneurons and the excitatory cells develop hand in hand. That's not the case in the dorsal organoids. So one idea that I have and others have had, Paolo Arlotta, is that those interneurons that emerge in the dorsal organoids might actually be more like the olfactory bulb interneurons, which do arise from progenitor regions in the dorsal cortex at later stages. And they're not cortical they're not cortical interneurons. They're interneurons that are destined for the olfactory bulb, but they're GABAergic and they express DLX genes. And no one yet has come up with a gene signature that I'm aware of that distinguishes those progenitors or those early GABAergic olfactory bulb cells from cortical interneurons. So I'm wondering, haven't proved this, whether those really are cortical interneurons. My guess is they're probably not, because they're just not coming from the structure that normally produces cortical interneurons in developing brain. That's a long answer to your question, but that's my interpretation.
No, thank you. It was super clear because I'm never sure with these organoids whether you have these different structures. So I know there are some interneurons coming up, but I'm not really sure whether they are like the ones you would get in normal development. So it was super clear. I think I saw those papers where they combined the two types of, and they can see migration from one to the other. Yeah. They're really interesting.
Yeah, that wasn't very robust migration. I think they make bolder, like a lot of organoid papers, they make bolder claims than the data would normally support. But I think it's a good idea and it seems to work reasonably well.
Okay. Any other question? I can make some
Javier, you can unmute yourself. I'm happy to.
Yeah.
Javier?
Thank you, Javier. Yeah. Thank you very much. Amazing talk. And Alejandro said, as Alejandro said, it was a lot of data, and my mind is like exploding. And I will try to put something, into a question. The thing is that you are thinking that there is a proto-map of the neuronal side of the story. And I start to think that maybe there is also an astroglial proto-map, because the rest of the progenitors that are not going to be neurons, they're going to be astrocytes. So maybe the differences among the areas of the brain that we saw in astrocytes is due to the proto-map. I mean, or I am, my imagination is going too far. Please give me some light on that.
Yeah, no, you're absolutely right. That's really interesting. So our fetal tissue, unfortunately, doesn't capture stages of, really significant astrogenesis. Most of the glial cells are produced in the third trimester, and the tissue that we get that we study that are donated samples are usually in the second trimester. We don't get third trimester samples except very rarely from the neonatal ICU postmortem tissue. So we haven't really done as extensive a study of gliogenesis as we have of neurogenesis. But if you look at the Allen Brain Institute data for the adult human brain, they have a lot more regional identity in astrocytes across the cortex than you see in, say, oligodendrocytes or microglia. So it's clear, as you suggest, that the astrocytes do have sort of address data. They have specific addresses, and they're different in the frontal cortex than the occipital cortex, and more so than you see in other glial populations like oligodendrocytes or microglia. And my guess is that it's going to be very similar to what I showed you in the Proto-Map in neurons, namely that especially differences between the poles, the frontal pole, the occipital pole, I suspect those astrocytes are going to be very different in identity, and the Allen data supports that idea. But even in between, those areal differences do occur in the astrocytes more so than other glial cells. Now, whether that has anything to do with their function, if the function is somehow different in those different brain areas, is I think unanswered question, but it's an intriguing issue. And of course, there could be disease relevance too. There could be certainly vulnerable astrocytes in certain regions, more so than other regions, in diseases that are associated with inflammation or with changes in reactive astrocytes. I'm just guessing in that case, but there are genetic differences that may underlie that.
Thank you very much.
Okay, I have a couple of questions. So one is, it looks like in one of the movies that you show, the radiclia migrates like in kind of interkinetic migration, but without reaching the ventricular area. So it goes, it divides only in the close to the basal membrane. Go to the. Is that the case? Because in other movies you show different kind of behavior. Right. That they migrate close to the.
So most of the time, they migrate toward the cortical surface. So they go from some, sort of basal to apical direction. Oh, sorry, from apical to basal direction.
Yes.
And they have a fiber that's a basal fiber, and they jump along that fiber. But every so often you'll see a cell going in the opposite direction. It'll jump in the opposite direction and then divide. And that's something that has been seen in slice cultures of non-human primate. Henry Kennedy and Colette DeHaye published studies where they did time-lapse imaging, like I showed you in human, in macaque and visual cortex at early stages, and they see cells jumping in both directions. So we look more carefully, and we think the cells that are jumping in the direction of the ventricle are not, maybe not always, but are certainly many times not neural progenitor outer radioglia, but are outer radioglia that are making that will be making oligodendrocytes. So we think there may be differences in the direction that actually have to do with different types of progenitors. It's not the same outer radioglial cell going and jumping up and jumping down, as it were. And we also see cells migrating along those fibers that are also dividing, and those progenitors that are migrating up and down are definitely oligodendrocyte precursors. We've labeled them, and we have a paper on that you could take a look at with time-lapse images showing highly dynamic behavior of these progenitor cells, which are in the lineage for oligodendrocytes, not for neurons. So we think there's quite an interesting diversity of behavior that relates to the progenitor type. And when it comes to interkinetic nuclear migration, that's really confined to the neuroepithelial cells and the ventricular radioglia, that oscillatory behavior that occurs as they pass through the cell cycle with division at the ventricular surface. That seems to be pretty exclusive to neuroepithelial cells and ventricular radioglia, and also the truncated radioglia that I mentioned later on.
Great. And then the hippocampus also have this feature that is similar to the outer radioglia. So do you kind of connect these two kind of cells?
Right. So we haven't done a lot of studies that are focused on the hippocampus, although now we're collaborating with others who are very interested in the hippocampus. What I can say is that there are HOP-X positive cells, which we use as a marker for outer radioglia, that are in that, the granular layer in the hippocampus that is thought to be a site of adult neurogenesis. So, we're very intrigued about whether there could be outer radioglia-like cells in the human hippocampus, but so far it doesn't look like it. It just looks as though the gene expression patterns for the radioglia in the hippocampus are a little bit blended, it's a little unclear whether you could draw a direct line between the truncated radioglia, the outer radioglia, or those, radioglia in the so-called adult hippocampus. So we're not sure yet. And I think it can be a little misleading to just focus on markers, individual markers. I think you have to look at other features of the cells if you're trying to classify them. So I think there's more work to be done in looking at at least the human hippocampus and sorting out the different progenitors. And I should mention, my colleague Arturo Alvarez-Bulla at UCSF has been very interested in that. And he's shown, among other things, that the progenitors disappear in the hippocampus as the human brain approaches adolescence around 10 to 12 years of age. So it's very different than the mouse, where those cells continue throughout life. So there are already some interesting differences, but I think there's more work to be done to sort that all out. And
in your kind of this proto, I didn't get the term.
Protocortex?
Protocortex is more like the temporal ones. The hippocampus will be in the temporal,
yes,
kind of shift in the, okay.
That's right. Although, we haven't looked at in detail, but that's exactly what one would predict.
And then, do you know if the glioblastoma cells have the leaf expression? The expression of the receptor of leaf?
Yes, they do.
They do. Okay, that's
right. And that's interesting, as you're suggesting, because it's a self-renewal pathway, and it suggests that it would be something you'd want to inhibit as a potential therapy for or a way of reducing the invasion of the tumor.
Then for the organoid story, some people kind of argue that the cells do not mature. The cells that are derived from these induced pluripotent stem cells, there are difficulties in maturing. So then you don't get the completely differentiation of the cells, complete differentiation. So would you agree with this hypothesis, or could be due to this stress ER stress and glycolysis, increased glycolysis.
Yes, well, I'm sure that the stress is, negatively affecting the ability of these organoids to really match what happens during normal development. So there are reasonable approximation of what happens during normal brain development, and they're very useful in many ways for studying things you couldn't approach in other models, especially in the mouse. But you just have to be cautious about over-interpreting the results. And what you mentioned about maturation is very interesting, and differentiation. We can see that there's a kind of distortion in the maturation program in specific cell types. And others have also noticed this, that as the organoids get older and older, they don't always mature. They don't continue to get features that develop as normal cells would mature. Some of them are arrested at early stages and never go much beyond that in terms of individual cell types, like certain excitatory neurons, for instance. Glial cells, astrocytes, develop in organoids only at later stages, as they do in normally developing brain. And once again, there's work, I think, from Sergio Pasco's lab that shows differences between early and late stages of astrocytes. When I say late, it's over about a year-old organoids, when they start to show gene expression patterns that you see after birth in astrocytes in humans. And I should mention the early astrocytes look a lot like the outer radioglial cells in terms of their gene expression patterns, because they derive from outer radioglial cells. And if you grow them long enough in organoids, you start to see the outer radioglial genes going down and the more mature glial genes coming up. But there again, they never look quite like mature astrocytes. Nobody has actually gotten them to look really mature. And that's one of the problems. People have gotten protocols to make microglia and astrocytes directly from pluripotent stem cells in a short period of time, like 15 days, essentially a little over 2 weeks. And those actually look more like the mature versions of microglia and astrocytes than these year-old organoids. So organoids are not always the best way to make a particular cell type. And I think people have to adopt the in vitro model that fits the question they want to ask. It's not maybe necessary to make every cell type in an organoid that looks like a brain if you're studying microglia. You might be much better off just making microglia in a different way. So these are all fascinating models, and I think people are just beginning to understand their limitations. But I think you have to be careful. People, it's much easier to publish a paper with a fancy claim, and then later on, one sort of backs off and decides it's not quite what they thought it was originally. I think that's natural, normal, but it's happening a lot in the organoid field.
Great. Thank you, Arnold. Any other question? Any other? Okay. So it seems that we have no more questions. So great talk. I mean, all the information that you have given us, it's amazing. I mean, all of these different results, and so we have to kind of process all of this data. But it's a very, very nice input to our knowledge. So
well, thank you, and I appreciate everybody who tuned in, and good luck, all of you, especially the students and postdocs. Hopefully they'll do great things, and we'll be reading about it soon.
Yeah, and hopefully maybe in person.
Yeah, that would be great.
We can invite you. Yeah,
that'd be terrific. Thanks again, and
thanks a lot.
Take care and stay healthy.
Bye,
Thank you very much.